Hi, everybody, and welcome to Neuroscientists Talk Shop, UTSA's Neuroscience Research Podcast. Today is April 4th, 2025, and we're talking with Punam Pokam, who is Assistant Professor in the Department of Neuroscience and Experimental Therapeutics at the Texas A&M School of Medicine. Punam's lab works on mechanisms controlling brain network excitability, including changes that accompany excitability disorders like epilepsy and traumatic brain injury. Welcome.
Hi. Thank you so much for having me here.
It's good to have you. I'm Charlie Wilson, your host. So, Punam, big changes in brain activity produce big changes in osmotic regulation in the brain, and phenomena like epilepsy and some of the features of traumatic brain injury and spreading depression and depolarization are all associated with changes in extracellular space and cell volume. So could you start by explaining the cellular mechanisms that control cell volume and the movement of water across cell membranes, and then you start talking about how they change in some of those conditions?
Definitely. So in terms of traumatic brain injury, we know neurons, glial cells, they do swell after traumatic brain injury. And that occurs a few hours after the injury. And that can, the swelling can continue for days after injury. One of the mechanisms...
You mean stay or getting worse and worse?
That's we don't have any continuous monitoring of neuronal or glial swelling. What we have is the snapshots. Uh-huh. So that's one of the study that we are actually conducting as of now, is doing chronic monitoring so that we can understand, is it a progressive increases, or is it just a transient increase, but over time it's kind of gets reduced. Uh-huh. So that question we still don't know, but hopefully with few studies that we are planning now, we'll get a clear picture of neuronal swelling and also the astrocyte swelling. One of the mechanisms that we know that can cause neurons to swell is the changes in the cation chloride transporters. These are mainly sodium and potassium driven NKCC1 transporters and KCC2 transporters. And we know after TBI, they are also altered. So their expression can alter. But there's also a way that can influence the conductances through NKCC1, causing neurons to swell. So these are the main mechanisms that we know in terms of neuronal swelling. For astrocyte swelling, aquaporin 4 has been well-known contributor for swelling in glial cells. And we also know that after TBI, expression of these aquaporins are altered, causing astrocytes to swell. So these are primary mechanisms that can contribute to cytotoxic edema after TBI.
And the way these work is that they use the ionic balance between the inside and outside to drive the movement of water back and forth.
Exactly. So one way you can think about it's driven by sodium and potassium permeability, or the concentration in and out of the cell. But there's also other way is just the changes in the transporters number. If they're not pumping out the water enough, that can cause also swelling, right? So there are two things.
So it's active.
Active, exactly. And so when I say about is it driven by changes in the sodium and potassium? Yes. If there are increases in the potassium in the extracellular space, that can drive more conductances through NKCC1, irrespective of changes in the channel or transporter numbers. And if the extracellular potassium goes up, NKCC1 conductances can increase, causing water molecules to get into the cell, causing neurons to swell. So there are two mechanisms that can go hand in hand. Yeah.
And then the ions, they are regulated independently by other things like neuronal activity.
Exactly. And also the vascular changes, right? So we know after TBI or traumatic brain injury, we know blood-brain barrier permeability is affected. And what that's going to do is pump potassium from the blood vessel into the interstitial space. And that can cause increases in potassium, causing NKCC1 conductances increases in neurons. So that's one pathway. The other pathway is, say, for example, neurons are just firing a lot. And if they're firing action potentials consistently or their frequencies increase, during the repolarization of action potential, they're going to be pumping potassium into the extracellular space. In turn, there could be accumulation of potassium, causing again the conductances changes driven by NKCC1. The third factor could be astrocytes not doing their job of clearing potassium and glutamate from extracellular space. So if neurons are firing, releasing potassium, glutamate, and astrocytes are not taking up, not doing their job of clearing potassium and glutamate, that can again cause extracellular accumulation of potassium, in turn changes in the NKCC1 driving cell swelling. So there are multiple mechanisms that could cause neuronal
and they all
swelling.
interact with each other.
Exactly.
Changes in extracellular volume alter the potassium concentration and so on. It sounds like you can easily get your brain in trouble from an osmotic perspective. So do we know what the consequences of that are? I mean, obviously, some part of the brain in traumatic brain injury is actually damaged, directly damaged. But then the spread of that is partly, maybe mostly carried by these kinds of changes, or
Yeah. So what we know, because I don't want to neglect the literature that existed, in non-TBI, non-traumatic brain injury brains, because some of the experiments were also done by postdoc mentor Ed Dudek. So what they showed is when you change the osmolality of the extracellular solution, like ACSF, in terms of the solution that we use, if you change the osmolality, if you reduce the osmolality, what you get is this synchronous firing of neurons. They're just crazily firing bunch of action potentials. So you get something like seizure-like activity in brain slices if you change the osmolality.
So how does that happen? What's causing that?
Yeah, so it's I think the mechanism or the hypothesis that they had is when you are changing neuronal or astrocytes to their size, that changing the extracellular space. And what's happening is now neurons are coming physically close to each other, kind of, and they can just passively conduct the action potential currents because they are so close by. And that's what they call emphatic interaction that could cause a seizure-like interactions or seizure-like discharges. And if you change the osmolality, if you increase the osmolality, you can reduce the seizure-like discharges. So that's what, some of the mechanism we know in the non-TBI brain. But what we found out a few years ago when we tried studying this, what neuron swelling is doing to the brain excitability, what we found is when neurons are swollen, there's actually decreased excitability of neurons. Their intrinsic excitability is decreased. And it's also on the network level, they are less excitable. So network excitability is also reduced.
So that's something. The extracellular space presumably is shrunk.
Yeah.
And the cells are really close together, but for some reason that doesn't cause them to be overexcited.
Yeah, but that's also one thing, all the emphatic interaction mediated synchronous firing following osmolality changes, they were done in hippocampus, where we know the architecture of the CA1, pyramidal neurons, they're physically close to each other, right? Their neurons are just packed, right? Yeah. Cortex, that's not the situation. So we are recording in layer 2, 3 somatosensory cortex. And one of the reasons could be we are seeing this effect, maybe it's just a region specific, right? So that's what I think, because, unless I do recordings in the hippocampus to show that there's also reduced excitability, I'm not going to just say that, I do expect the same kind of changes or reduced excitability in the hippocampus.
So do we know, not worrying about that, but just thinking about the phenomenon you're seeing.
Yeah.
What do we think is causing that reduction in excitability?
Yeah, I think it's to do with the cells, like the neurons swollen. They have now their capacitance, the membrane capacitance is increased. And one hypothesis that we had is like there are now more receptors or channels that can conduct the currents. They are more in numbers now after TBI, just because of their size increases. And if they are conducting more, their input resistance is going to be reduced. And what that's going to do, if there's a less input resistance, you need more currents, any synaptic currents, to make changes in the voltage in the cell at the cell level. So this intrinsic excitability reduction is what's causing the network to be less excitable. That's what we think is.
So I know you've measured the intrinsic excitability a lot, so you can answer the key questions about that for me. Like one reason a cell might become less excitable is that it just has a more negative membrane potential. So threshold. So can you tell us what happens to the membrane potential?
That's a great question. So we did not see differences in the resting membrane potential. We also did not see changes in the threshold for action potential firing. So because that's one of the things that we hypothesized initially, that even though we are recording in the area that's far from the injury site, these supposed to be the spared neurons, right? The spared cells, which should be functionally active. But what if they are also just, their membrane potential is so depolarized that now they are not going to be excitable anymore because they are close to the universal potential.
Depolarization block.
Exactly. So we did not see that difference between resting membrane potential between sham and TBI. So
you still see neurons firing normally. They're not in depolarization.
Yeah, they are only firing less low in numbers, but there was no differences in the threshold for action potential firing. Yeah.
So what are the other measurements we use when we don't see membrane potential or threshold changes? We look at the input resistance. Mm-hmm. So a neuron has a small input resistance.
Yeah, so input resistance, after TBI, they were reduced. And so one can say that, just by looking at the Ohm's law, it's like simple Ohm's law, V equals IR. If the input resistance is less, the voltage changes are going to be smaller also, right? And so,
so it just takes a lot more synaptic input or a lot more current.
Exactly.
To make the cell fire.
Yeah, so that's why they are less excitable. So even though the input coming in is maybe the same for sham and TBI neurons, but the voltage changes are going to be just smaller for TBI. They need more, double the input that sham neurons will receive.
So that is like double? It's
like, I'm making the number, but something like higher the synaptic input that they need in order to make similar voltage changes.
So do we think this is a good thing for the recovery from, because the idea, I think, I mean, you correct me about this, but I think the idea is that there's a damaged region in the center, and then there's a region of brain that's at risk around there. And that part could go either way. It could die or it could recover. And the trick is to try to get as much of it to recover as possible. So now if we know that part of the brain is getting quiet, or is it the whole brain, or does it have to be near the damage to go quiet like that, or is it the whole brain?
Yeah, no, that's a great question. What we recorded is almost like 1 to 2 millimeter far from the injury site.
So that's pretty close still.
Yeah, still it's close. But I think we are also now doing a little bit further from the injury site to see is there kind of like a graded area, right? Like close to, say, if it's 1 to 2 millimeter, even we are talking about the mouse brain, right? 1 to 2 millimeter, you have hypoexcitability. But what if if you go a little further, they have something different? Or is it it doesn't matter?
Or maybe you could be looking in the thalamus or in the red nucleus, the whole brain is kind of decreasing its excitability.
So that's again the great point. We are also now recording in the thalamic regions, BPM mainly, where we want to understand how that region is affected after TBI, whether there is based on literature, we know after TBI, weeks later, there is like a neuronal loss in the thalamus. But there's also like, not chronic recordings to show are there like immediate changes, or there's really nothing happens until a week later, the injury, that thing starts like, dying. Or the cell starts dying.
So that
and it
could just be
the synaptic input coming, synaptic input coming from the cortex is less, and so neurons are not receiving the synaptic input, and they are just eventually dying.
Or even the thalamic neurons no longer have a target.
Oh yeah, exactly.
And so degenerate in the retrograde direction, which thalamic neurons are famous.
Yeah, that's a great point. And so that also brings back to my next point, that we also need to look at like, layer 4, which is like layer 4 of the cortex, which is like more the recipient of thalamic input, how that's affected, and also layer 6, which sends out the like output to thalamus, how that's affected. So like, we still don't know how like, these cortical layers, how they are altered. Different layers are altered after TBI, and how that contributes to the subcortical regions. Yeah.
So to evaluate whether this change in excitability is something we ought to treat people for, or something we ought to be happy that it's happening, and leave it alone, because the question is that promoting the destruction of brain tissue, or is it protecting against the destruction of brain tissue?
So that's like, it's a great question. So I have talked to few of the neurosurgeons, okay? So one neurosurgeon, he was very excited. He's from University of Utah. He was very excited when he read our paper where we showed the neuronal swelling needs to be conserved. We need that protection, because if you reduce the neuronal swelling, everything is going to get chaotic. You are going to get increased excitability, which you don't want it, presumably. You don't want seizures. You don't want like, spreading depolarizations like event. So what he was interested in kind of like mimicking our study in the severe TBI, traumatic brain injury model, because most of the patients that come to the ER, emergency department, it's mostly like, they have severe traumatic brain injury, not the moderate TBI injury that we studied. So he got excited because there is also like, he has seen clinical patients. He has seen some like, literature where even though you try to reduce intracranial pressure following TBI, those patients do not do better over time. They actually do worse.
And the intracranial pressure is caused by the brain swelling.
The brain swelling,
this
exactly.
Is two sides of the same coin.
Yeah, exactly. So that's why he was very excited. He's like, maybe the findings that you have, they have some clinical implications. So that is something that we have to continue in order to understand, because you don't want the increased excitability like seizures and spreading depolarization. But what about like, if there's a hypoactivity, what it's doing to the learning and memory? If there's a reduced activity, like, are we saying that the plasticity, or like, plasticity, which is one of the mechanisms for learning and memory, is it also affected? If that's true, then we don't want to protect neuronal swelling. Like, we don't want to have neuronal swelling.
Well, how long is the danger area? So I'm kind of picturing, been some damage, and now there's a damaged place. There's probably no bringing it back, but there's a region around this that's now at risk, and the damage could spread to that region, or it might not, or it might spread a little ways and then be checked by whatever natural processes check that. And then what happens? I mean, is everything going to reach some kind of steady state, and you can quit worrying about whether the brain is swelling or not swelling because it's going to go back to normal, the part that doesn't die? Or is this a brain swelling something that's going to go on forever after this event?
Yeah, so in animal models, what we have seen is like 7 days post-TBI, everything recovers. So cell swelling.
If the brain just shut itself off and didn't learn anything for 7 days, but that caused it to recover, it wouldn't be a high price to pay. Yeah,
but the thing is, what if the changes that are happening at 48 hours because of the brain swelling, right? If they're worse or good, if you're not taking care of that time, you are going to see changes at 1 week post-TBI. So that's another project that we want to do, is like, when you protect neuronal swelling early on, what are the long-term effects? And if you don't treat neuronal swelling, that we have seen, like, not treating neuronal swelling, and 7 days everything recovers, but the excitability is increased.
Uh-huh.
So it's like, we need that long-term recordings where we understand this, like, swelling and excitability relationship.
Yeah.
The other thing is, like, the point that you made is, you have injury, and then you have this kind of like at-risk area around it, right? You are envisioning this as, like, stroke, where we have a lesion and then we have,
I'm
like,
thinking about
right,
exactly
like a stroke.
the same as
Yeah. But one thing that always, like, amazes me is, like, in the stroke, we know the infarct area, the middle, what's, like, all damaged. The spreading depolarization, like, event, when it occurs there, it propagates to, like, those spared areas, right? But then there would be spreading depolarization coming from the spared area towards the lesion. Okay?
Yeah.
But in case of traumatic brain injury, whenever we have recorded spreading depolarization, we have always seen spreading depolarization propagating from the injury site.
One way only.
One way, and that's it. We don't see spreading depolarization for like 5 hours, 6 hours. But there's also, like, we don't have long-term monitoring of sodium spreading depolarization after TBI to know, is it like one hit, or is it like, we get multiple over days maybe. So that always amazes me, like, why one spreading depolarization?
Now we have to talk about spreading depolarization. We should.
Yeah, definitely. So let me just give, like, background on what is spreading depolarization. So spreading depolarization is the excitability phenotype that has been seen in epilepsy patients. Like, it's a clinical phenotype that has been seen in epilepsy patients and also traumatic brain injury patients. And in animal models, what we know is when you induce spreading depolarization, you can induce spreading depolarization if you increase the neuronal firing, if you increase extracellular potassium, if you increase glutamate, you can induce spreading depolarization.
And what's this look like?
Huh?
How do we detect spreading depolarization?
Yeah, so spreading depolarization, so once it occurs, it's kind of like, if you are recording from single neuron and you are inside the cell, your whole cell patch clamped, and you're recording membrane potential, what you see is the massive depolarization that occurs. And the magnitude of this depolarization can be like 50, 60 millivolts.
So the change loses its membrane potential.
Exactly. But then it does recover in, like, 2 minutes or so. Then if you are, like, in the extracellular space and you have, like, just the electrode recording local field potential, what you see is the large negativity in the extracellular space, like, in the potential.
With the same duration.
Same kind of, like, 1 to 2 minutes duration. Yeah. If you record astrocyte membrane potential, you also see that kind of, like, massive depolarization, and the duration would be also longer, like, 2 minutes. So this spreading depolarization, let me compare it to, like, seizures, which we all know how the seizures are, like, how kind of like crazy activity we see during seizures. It's like lots of neurons are fighting continuously, right, during seizure activity. But here in the spreading depolarization, what happens is at the initiation, like at the start of the event, you see some neurons firing, like, you see some action potential, but then neurons would just depolarize, that there won't be any action potential firing or anything because it would be close to the,
the block.
And then it would return back, like, after 2 minutes. But that during that whole period, it just, the cells are depolarized, right? The neurons are depolarized. So that's what you would see at the cell level. If you record potassium or glutamate in the extracellular space, what you see is, again, the dramatic increases, like potassium can go up to like 40 millimolar in concentration. In contrast, during seizures, we know it can go up to like 6 to 10 millimolar or 12 millimolar increases in extracellular potassium. So you can see the magnitude of...
This is an extreme event.
Exactly. It's like, it's so extreme event. But even though now people have been realizing that it's not just sparing depolarization, shunting, or shutting down the neuronal activity, there's more to it. What I mean by that, so in epilepsy literature, if you talk to anyone doing epilepsy, if they are studying seizures in slices, what they see is bunch of seizure activity, and then you would get this sparing depolarization event, which would cause post-ictal depression, and that can last for 20 minutes.
The brain just goes quiet.
It's just depression compared to the background activity that's seen before seizure. So if you talk to an epilepsy person, they would be like, oh, it's just terminating seizures. Okay, because what's happening is you get these seizures, and then seizures would kind of culminate into sparing depolarization. So when you look at just when you filter out this DC recording or the negative shift in the field potential, what you see is a flat line, falling seizure.
The flat line isn't just the absence of a seizure, it's a flat line.
Yeah, so what people always say is that it's terminating seizures, so it's good. Because we want to terminate seizures, right?
Yeah.
So it's a good thing. What we don't understand is the tissue, if it's going through sparing depolarization, that means there is inherent or there is intrinsic hyperexcitability that's increased, or the excitability is increased to the level that now it's going into the sparing depolarization event. And we also know sparing depolarization can cause tissue damage, histology, if you do a bunch of histology, you can see, there's more reactivity of the astrocytes, there's more damage, cell loss. So it's not just terminating seizures, it's actually doing more than that.
Yeah.
And I think that's what, I think...
it's the ultimate excitation. So if there's excitotoxicity, that's when it's going to happen.
Exactly. It's yes, it is terminating seizures, but you are getting this massive event that you don't want you to have, right?
So I have a bunch of questions about it. I don't want to just end up asking a bunch of questions, but I do want to ask a bunch of questions. So one is, if you just dump a bunch of glutamate on neurons, they'll depolarize very powerfully, up to 0. Mm-hmm. And you could think, oh, that's all there is to it. But that doesn't happen to astrocytes. They don't just depolarize up to 0 when you dump a bunch of glutamate on them.
Yeah.
So what's the relationship between glutamate and neurons and astrocytes? Can we kind of put together a sequence of events that causes this giant depolarization? And then, if we can do that, what stops it? Why didn't it just stay depolarized forever?
Yeah. Good questions. So, one of the experiments that our PhD student, when he was trying to do, we were working on the sequence of the sparing depolarization. One of the experiments that he did, he actually he was doing brain slice electrophysiology. He was increasing glutamate concentration, and he patched neuron. And just by increasing glutamate concentration, what he saw was neurons were not actually depolarizing to that extent. There were actually some neurons would have upstate-like event. The cells would depolarize, would come back. Sometimes they would have action potentials. But there wouldn't be greater response in neuronal depolarization following changes in the glutamate concentration. In contrast, if you increase potassium, neurons would depolarize. You would get that graded response. And when he showed that, and that's we knew it based on experiments where people have done just the glutamate glutamate puff and trying to induce STs. It's not sparing depolarization. Sorry, I keep saying STs. They were not able to induce sparing depolarization. But if they block glutamate transporters, Like, astrocytes have glutamate transporters, neurons have. If they block that, then they can induce sparing depolarization.
Because then the glutamate just hangs around in every
Exactly.
spit.
Because what I think is in the brain slice or in vivo, when you are puffing glutamate or when you are trying to increase glutamate in the extracellular space, these transporters are very active. They are doing their job. So there won't be enough glutamate that could cause sparing depolarization. So that's one of the things that I...
So when you see this happening in a traumatic brain injury or near a stroke, then what do you think happened? I mean, maybe it was glutamate that caused the potassium release in the first place or something?
I think it's a
but it's
chicken and
really
egg.
Potassium. It's really potassium because without the potassium you couldn't get.
Yeah, so it's actually a very chicken and egg question. Who comes first? Is it potassium or the glutamate? So the techniques, so the experiments where we recorded extracellular potassium with potassium sensitive electrodes. Then we recorded glutamate with the genetically encoded glutamate indicator. Then we recorded astrocyte membrane potential, which we know can be used as a potassium electrode, right? Because astrocytes depolarize, astrocytes can depolarize any smaller changes in extracellular potassium. So they are very well-known potassium indicator. So we did a bunch of experiments where we were recording astrocytes membrane potential, extracellular potassium, then extracellular glutamate changes during just sparing depolarization in the naive animals. And what we saw that these astrocytes, their potassium increases or astrocytes membrane potential changes that precedes the sparing depolarization event. And the potassium increases in the extracellular space, that changes occur at just 5 seconds before the sparing depolarization. But it does occur before glutamate increases. But one can argue that the indicator that you have for glutamate recordings, maybe it's not sensitive enough, that it doesn't record small changes.
Yeah, when everything's happening at once, it's kind of hard to find out who's the leader.
So there is 5 second delay between potassium and glutamate. And if you think astrocyte membrane potential is the ultimate potassium electrode, these membrane potential changes are happening almost 20-30 seconds before sparing depolarization. So if you think it's only extracellular potassium changes causing astrocytes to depolarize, then one can say it's a potassium driven. But I don't buy that. I think it's a glutamate.
In experiments, people often just add some potassium, or dab a little potassium on the cortex and start a depression of it. But in damaged brain tissue, why do they happen?
Because I think the injury that we are inducing, there's lots of damage. Cells are dying, and they are just dumping glutamate, potassium, and that we know there could be local increases in potassium, and that's it's a perfect trigger to induce sparing.
So that would explain why there would be a generator at the site of damage.
Exactly.
And not out at the edges. So now, I don't want to fail to ask you about what stops them. So if you can make these measurements of potassium going up and glutamate going up and so on, then and membrane potential going up, then you can also make them on the falling phase of the depolarization too. And what do you see there?
Yeah, so the glutamate changes, they even though they occur at the time of sparing depolarization, they recover within 20 seconds, way before neurons membrane potential recovers. Neuronal membrane potential recovery is kind of relates to what happens in the extracellular potassium recovery. So the timeline, the time that it takes for potassium to recover, it's kind of the same for the neuronal membrane potential to recover.
So some kind of potassium clearance mechanism is kicking in and knocking this thing down.
Yeah. So one hypothesis that we always have been working with is if you make astrocytes more functional of clearing their, clearing extracellular potassium and glutamate, can you delay or can you reduce spine depolarization happening?
So one thing you said earlier was if you reverse the swelling, that those spreading depolarization events will be more frequent.
Yeah.
So do we have a clue about why that would, how the swelling is connected to the generation of those?
Yeah, so I think swelling, one thing you would expect is when the cells are swollen, the extracellular potassium, I think we talked about this when the extracellular potassium, the concentration can be increased just because the extracellular space is now shrunk, right? Because the cells are swollen. In that case, what you would expect is the local neuron hyperexcitability. But it's also if the extracellular space is shrunk, you can imagine if there is spine depolarization happening, the diffusion, the propagation would be limited just because of the extracellular space being limited.
Because it's spreading because potassium is
diffusing. Of course, there is also intercellular pathway of the astrocytes that can also cause the propagation, but I think it's mostly also the neurons, neuronal depolarization spreading, and also it's also potassium diffusion to the extracellular space, causing spine depolarization to spread. But when you reduce neuronal swelling, increases in excitability and also increases in number of spine depolarization event. It's if you don't have that protective effect of cell swelling, if you take away that protective effect, now the intrinsic excitability is going to be high. The cells are going to be more intrinsically excitable. And you can imagine if there are a bunch of neurons are more hyperexcitable, they're at that threshold. So any trigger can drive
start the explosion and the potassium exposure,
and they can also have more number of spine depolarization just because the excitability is now increased.
So spreading depression events, like spreading depolarizations, which are also
cause spine depression,
and have been studied for years, and those things propagate over the entire cortex, not just in little animals' cortex, but in human cortex all the way along. So in traumatic brain injury, when those things are happening, do they go everywhere in the cortex like that? So I kind of wonder, because occasionally people have postulated that migraine is connected to spreading depression somehow, and I know that we don't have time to go through that whole story, but I wonder if you could know what it feels like to have spreading depression, if one of these events were to just happen to me right now, somebody was secretly putting some potassium chloride on my cortex, and this wave of depolarization and depression went through my cortex. Would I just go unconscious during that, you think? Because my cortex would just go quiet, my EEG would flatline and for 20 minutes, and then it would recover. Do we have any, I'm not asking you to make up something.
No, I think it's a great question. So in migraine, I know when spine depolarization is also underlying mechanism for migraine with aura. And I know migraine aura, my colleagues have migraine aura, and they have of course they must be getting spine depolarization. We know because clinically people, there's a bunch of studies that show that when the patients have aura, and if you record their both signal, right, the blood oxygenation signal, they mimic spine depolarization signal. So I know it's happening.
So maybe you wouldn't go unconscious. Possibly you would just see hallucination or something.
Yeah, exactly. So one thing that I remember my colleague telling me, whenever he has migraine auras, he would be kind of stupid for 24 hours. He wouldn't be like, smart. And he's like MD, he's like very smart person, but like he would be kind of like, just like
sleepy.
And like he wouldn't be like, sharp.
Yeah.
That's what I've heard, but it's also very like, subjective. He may be analyzing more for himself.
Sure.
But in animal studies, because clinical studies where they have recorded spinal depolarization, these are also like, patients who have traumatic brain injury. And these recordings were done within like, 10 days of their like, when they are admitted to the ER. So that time, most of the patients are they're unconscious. So it's like, I don't know if there is any data, or maybe I'm just missing on the clinical data. But in animals data, we know that there's actually recent publication that showed that when there is a spinal depolarization, that can actually increase the activity in animals. There will be more like, the movement would be more.
That's remarkable. It's remarkable because your cortex would be completely flatlined and you're running around.
Exactly. But if you look at like, epilepsy literature, what we know is when the spinal depolarization cause that's associated with like, depression of the, it's associated with post-ictal depression. What we know is like this post-ictal depression can be associated with like, immobility in patients, in epileptic patients. So
which is what you'd expect, really.
Exactly. Yeah.
Yeah, I would think if my motor cortex goes flatline, I'm probably not going to be moving around.
I wouldn't say flatline, it's like it's reduced. But yeah, if it's reduced activity, you would expect like, less mobility or less activity, like motor coordination, not like, hyper.
Yeah.
Yeah.
Well, thanks a lot, Punam. This has been really fun talking to you.
Thank you so much. This was really great. This is the first time I'm doing it, but it just felt awesome.
Good.
Thank you.
This has been Neuroscientists Talk Shop.